Once a polypeptide's been made inside of a cell, there's a little bit more work that needs to go on in order for it to become a protein. So we see four main levels of protein structure that are required in order for us to turn a polypeptide into a fully fledged protein with a specific shape. So what we'll see is not all proteins will go right up to a fourth level of structure, but they need at least a couple levels of structuring in order for them to get the shape that they need to do their role. So without a particular structure and shape to it, polypeptide can't actually become a functioning protein. So the first level, the primary level of protein structure, occurs or involves the specific sequencing of amino acids in a polypeptide chain. So this picture here shows number 1 up the top. So at the end of translation, what we've done is we might have built a chain of amino acids where we might have a leucine, arginine, phenylalanine, and so on. So this sequence of polypeptides, specific sequence of amino acids in the polypeptide chain are what give us a primary protein structure. If we go down to this second diagram here where we see something called a pleated sheet and an alpha helix, so we've got sheets and helixes, two forms of secondary structure. What will actually happen is some particular attractions in the form of hydrogen bonds will occur between different amino acids. So for example, if we look up at our diagram at the top here again, say the structure of leucine and the structure of phenylalanine give rise to a hydrogen bond being formed, then these two amino acids are going to have an attraction to one another. So they're going to draw closer to each other and start to change the shape. So that's what's going to give rise to a polypeptide starting to look like a pleated sheet, perhaps. When it's got this corrugated structure. Or we can start to see the amino acids forming a helix like so. Because of the attractions and the hydrogen bonding between amino acids along the polypeptide chain, it will start to change its shape because they'll be moving closer to each other and forming attractions. So that's the second level of protein structure. The third level, or tertiary level of protein structure, pictured there, is once we've got a pleated sheet or a helix or a combination of the two, a polypeptide has formed a pleated sheet and or helix type structure, secondary structure, we can then see further attractions between parts of that structure which will form this three-dimensional bending and twisting so that we start to get a more complex and more specific shape. So there are particular sort of side chains of amino acids which we can call R groups that will start to form attractions between other groups, and this is what gives us a more complex structure. So a big worm shape starts to twist and turn and combine into a more complex manner. And down the bottom here, our fourth and last level of structure, a quaternary protein structure, is essentially where we get more than one polypeptide chain combining together and binding together in order to form a more complex protein. A good example of a protein with quaternary structure is hemoglobin. Hopefully I've spelled that right. Hemoglobin. So hemoglobin is made up of four different proteins or four different polypeptide chains that have folded at a tertiary level, and then those four tertiary folded proteins combine together to create hemoglobin, which is quaternary because we've got multiple polypeptides folded into proteins joining together. So the most important thing to note about this is this folding is crucial in the performance of proteins because they must have a certain shape, and without a certain shape, a protein is going to struggle to do its role properly or not going to be able to function in the manner it is supposed to at all inside of an organism.